Imagine this. Every day, millions of sterilized flies descend from aircraft over Panama in a relentless battle against a formidable, but nearly invisible enemy. It may sound like a science fiction plot, but this operation has been a critical part of the United States' decades-long strategy to protect livestock from a deadly parasite, the New World screwworm. This flesh-eating menace has wreaked havoc on animal populations, prompting scientists to develop an unconventional but highly effective countermeasure. Though this approach remains the best solution today, new advancements may soon render it obsolete. The New World screwworm, despite its misleading name, is not a worm but a species of fly found across the Americas. Its rapid life cycle, lasting just 21 days, allows populations to grow at an alarming rate. Female screwworms deposit eggs in open wounds of mammals, including livestock, deer, and occasionally humans. When the larvae hatch, they feed on living tissue, causing severe damage and, in many cases, death. During the 1930s, these infestations devastated the U.S. livestock industry, inflicting millions of dollars in losses. Farmers attempted various methods to curb the outbreaks, such as shifting breeding seasons to colder months when screwworm activity declined. They also scheduled procedures like branding and castration during less active periods. However, these efforts proved ineffective, as the parasite thrived in wild animal populations, making eradication nearly impossible through traditional means. The breakthrough came in the 1950s with the development of the Sterile Insect Technique, SIT, pioneered by USDA researchers. This method involved exposing lab-reared screwworm flies to gamma radiation, rendering them sterile without affecting their ability to compete for mates. Timing was crucial, applying radiation between 5.5 and 7.5 days into their life cycle ensured sterility, while maintaining their natural behavior. Once released into the wild, these sterilized males mated with fertile females, preventing reproduction. Since female screwworms only mate once in their lifetime, a single encounter with a sterile partner effectively removed them from the breeding population. The first large-scale test took place in Curacao, where 400 sterile flies per square mile were released. Within 10 weeks, screwworms were completely eradicated from the island. Following this success, aircraft deployed millions of sterilized flies across the southeastern United States, ultimately eliminating the parasite by 1966. But the challenge wasn't over. Despite eradication in the U.S., screwworms continued to migrate from Mexico and Central America, necessitating ongoing aerial releases to prevent reinfestation. Maintaining this effort indefinitely was neither cost-effective nor practical. In response, the U.S. and Mexico collaborated in the 1970s to extend the program southward, sharing the financial burden. By the 1990s, an even more strategic solution emerged: the establishment of a permanent biological barrier at the Darien Gap, a dense jungle separating Panama from Colombia. This narrow, 97-kilometer stretch of land served as the ideal containment zone, far easier to manage than Mexico's 220-kilometer bottleneck or the expansive 3,200-kilometer U.S.-Mexico border. Today, a facility in Panama breeds and sterilizes millions of screwworm flies, which are then deployed daily along the border. This ongoing effort prevents the parasite from spreading northward, safeguarding livestock across Central and North America. While effective, the program remains costly and labor-intensive, driving researchers to explore new alternatives. One promising innovation involves CRISPR-based gene drives. Unlike SIT, which requires continuous intervention, this genetic engineering approach introduces a self-sustaining mechanism that selectively sterilizes female screwworms. The altered genes propagate naturally through successive generations, eventually collapsing the population. If successful, gene drives could offer a more permanent and scalable solution, particularly for regions like South America and Africa, where containment is more challenging. However, despite the clear advantages, concerns remain about the broader implications of genetic modification in wild populations. While few scientists worry about eliminating screwworms specifically, gene-drive technology as a whole raises ethical and ecological questions that must be carefully addressed. For decades, the fight against screwworms has showcased the ingenuity of scientific solutions, turning the insect's own biology against them. While Panama remains the front line of this battle, emerging technologies may soon shift the approach from containment to complete eradication. If gene drives prove effective, we could see a future where these destructive parasites are eliminated entirely, saving millions of animals and preventing economic losses worldwide. The question remains, are we ready to take the next step in this fight, or should we proceed with caution when reshaping nature itself? Let us know your thoughts in the comments below.